It's a pleasure for me to welcome you to the second part of this exciting day where we all are learning from each other. And my privilege is to lead together with a distinguished faculty through the topic of AI in clinical trials, moving clinical trials into the future. My name is Nicole Selenko-Gebauer. I am the Chief Innovation Officer at Danaher Diagnostics, and it's a privilege for me as a clinician to partner with all of our operating companies to co-create innovation solutions to help us move and advance healthcare into the future with precision medicine. So a few notes that I've been taking from this morning that I do believe build bridges to our topic. We heard development makes 70% of development cost, of R&D cost for clinical drug development. We heard that things that don't work today will work tomorrow. We also heard that in discovery or translation, that it's sometimes the off-target effects that show efficacy. We also heard that the first generation of AI-enabled molecules will not work. We heard about the value of surrogate endpoints, in particular in diseases where targets are ill-defined or where it takes a long time to show that a certain therapeutic intervention has an effect. And we learned about how critical it is to ask the right question. We heard a lot about adoption culture, and I wonder if we could translate this adoption culture, which is a bit blurry and nebulous, more into process and governance. And maybe there is an AI tool that helps organizations in this. So together with a fantastic faculty, I've had the pleasure to spend the last weeks to exchange on how to package the topic of clinical trials for you. And the way we will do this is right target, right time, Jeff, right symptoms, Ziad, right patients, right design, right comparators, right sites, right controls. And if we have the time, which I hope we do, we'll speak a little bit about how data advancements build new analog challenges. So please welcome on stage a distinguished faculty, Kate Sasser. Kate Sasser is the Chief Science Officer of Tempus AI. She is an expert in AI and precision medicine, and before Tempus, she was heading the R&D organization for Genmab, and we share some past on some innovation at Genmab together. So welcome, Kate. Ben Liu, I learned a lot from you, Ben, is the co-founder and CEO of Formation Bio, which is an organization that is using AI and large language models to enhance R&D and help physicians take the right decisions. And we saw some data this morning as to how that works, and we'll get your thoughts about this. So welcome, Ben. Jeff Elton. Jeff is the CEO of ConcertAI, a large multimodal data and AI SAS solutions company providing research and development solutions with a big focus of oncology. So I learned a lot from you there. Before doing this, he was the managing director of Accenture's strategy and patient health and the COO and SVP strategy of the Novartis Institutes of Biomedical Research. So welcome, Jeff. And last, but for sure not least, Ziad Obermeyer. It's a pleasure for me to introduce you in this panel. Ziad is an associate professor and Blue Cross California Distinguished Professor at the UC of Berkeley. He teaches at the School of Public Health and is a founding member of the Berkeley UCSF Joint Program in Computational Precision Health. His research uses machine learning to help doctors make better decisions. His work on algorithmic racial bias has been highly influential in shaping how healthcare organizations and policymakers hold AI accountable. Welcome, Ziad. So, when we started to engage and prepare this topic, we immediately jumped into this classical late-stage, large and long clinical trials topic when Jeff said, wait a minute, In the early space, the solutions are very different than in the late space. So what we will do is we'll start throughout the life cycle of R&D. So Jeff, tell us about AI solutions and data in translational medicines as well as phase 1 trials.
So to be clear, the solutions in translation are actually probably younger than they are in late stage clinical development, just because of the nature of the trials are so completely different. And actually, I have to admit, I found the morning sessions actually really interesting because that pointed out we're actually creating rational tools for doing drug discovery and advancing new therapeutic entities into the clinic, but actually human systems are very complex in terms of how they can interact around a new medicine. So one of the things we're trying to do, whether you start calling this digital twin technologies or others, that start to inform and saying, can I simulate how that one particular medicine may actually perform in that human system? Most of the work, as you introduced, most of the work we do is in oncology, hematology, et cetera. So if a particular patient population, a real population, actually got exposed to certain drugs, you can actually have patterns of resistance that develop, and you can actually get different responses from different subpopulations of patients. Some of that you can actually start to predict. So what we're trying to do is using multimodal data, that term's come up throughout the meeting today too. In our world, that means actually full exome, transcriptome, and could include image data of different modalities, longitudinal clinical records, put that together in a way that actually can be representative of the ultimate population that you may be seeking to kind of get into the clinical study. We've done this now in several different disease areas. We made some announcements at the JP Morgan with kind of one very large sponsor where we're starting to do that. We're beginning to advance that more, and we're spending a lot of time developing the different model-based approaches that allow us to do that. I do want to emphasize, we think it's extraordinarily promising because right now that early phase study, the word serendipity was always used. It's an attrition-based model. When you look at it in the industry, attrition means I can't predict what's going to fail and what's going to survive. So therefore, I run the experiment, and it's sort of Darwinian. Whatever kind of passes through gets to go to the next gate. That's not really an engineering kind of predictive-based process, and we think it's capable to kind of get there. So,
can I just ask you on this? We heard today AI-enabled molecules will not work, the first generation. What's your take on this?
I don't think they're. I mean, we're going from, a lot of people were having this conversation even coming in here. The term drug hunter gets used, and the reason why that term gets used is that means certain individuals have become their own expert system. I mean, when I was at Novartis, we had a couple individuals, they had better scientific taste, and so therefore we used them to help guide those early stage programs. That doesn't mean it was serendipity, it was an act. They refined their skills to kind of get better at that. And so I think this notion that models can start developing, and it's probably not going to be model. It won't be one LLM. It will be models that will be working, actually can start informing some of those same characteristics. I'm super optimistic about that. We're already seeing some of those characteristics.
Yes. Thank you so much. Ben, I see you already.
I was going to say, maybe my kind of perspective on this is, it's not that they won't work. There's just not really enough evidence that suggests that they will have a higher probabilities of success relative to a quote-unquote non-AI discovered drug. And part of that, I'm sure resonates a lot with folks in the room, that a lot of where AI is making a huge impact in drug discovery actually are things that help you get to an answer faster. But to John's point around kind of taste, maybe kind of we make the kind of comment that a lot of the best AI companies are a bit handicapped by not having the best tastemakers and drug pickers. So whatever relative advantage they have around the AI kind of systems, and most of the, I think, cutting edge AI allows you to maybe think about what might bind to what better, or these kind of questions that might take humans a lot of time to get to the answer. But once a drug is nominated to be a good drug in the clinic, most of the qualities of these drugs all have the same kind of aspects that a good drug picking team will have kind of filtered out. So kind of the precise statement is like, there's no a priori reason to believe that a drug might have a higher probability of success being AI driven or not. And to make the argument, most companies use some sort of machine learning AI, regardless of whether they're these quote unquote AI companies. And maybe a core reason why we focused, a company on the strategy around building models that can run clinical trials and drug development more efficiently was born out of my experience as a computational biologist, we're trying to use AI and ML to predict what's going to work before you run a trial. Long story short, you really don't have the data sets you need to make the clinical stage kind of predictions, but you might have the ability to make like dynamic predictions around what binds to what.
And we'll get to that point. So it's almost like a new business model for retired drug pickers and drug developers to teach computers. Before we go into the flow, we stay a little bit, and I'm bringing up the term of surrogate starting point. Is the way we look at clinical gestalt of a disease the right way? Do we start clinical trials at the right symptom level? Ziad.
Yeah, thanks, Nicole. I'm going to start by making an observation about clinical trials that is so obvious that I know I'm going to sound dumb. But when you want to run a trial of a drug for Alzheimer's disease, how do you get your patients? Well, you find the ones with Alzheimer's disease or some early form of cognitive impairment. And I think that's one of those things that's become so obvious that we've forgotten how many bad second-order effects that has on clinical drug development. For example, it means that the universe of drugs that we can put into trials are only drugs that can reverse a disease once the disease has already started to set in, which is a hard task, and it's probably a much harder task than drugs that prevent that disease from setting in. And so I think one of the things that I'm most excited about in this intersection of AI and clinical trials is the ability to, as Nicole put it, have not surrogate endpoints, although we'll get to those later,
yes,
but also surrogate starting points. So rather than enrolling people with things that we can write down as rules and instantiate in enrollment criteria, we have AI systems that, for example, look through all of the head CT scans that I trained as an emergency physician. I've ordered a lot of CT scans. We order a lot of tests. Those tests results, the images are sitting around in the medical record system of lots and lots of hospitals. We also have data on who ends up developing different kinds of dementia. So it would be very straightforward to have an algorithm that predicts the emergence of all of these dementias from head imaging, from neck imaging, there's lots of imaging that might be able to predict. So AI predicts who's going to develop the disease, and that's your enrollment criterion. It's not a bunch of human rules. It's someone who has a probability of developing Alzheimer's over the next time window. And this is all very incentive aligned, because you're going to see in your control group if your algorithm is good. But you're going to see in your treatment group if your drug is good. And so I think that could really generate a whole new kind of clinical trial that is geared towards prevention rather than treatment of diseases. It also opens up a bunch of interesting new opportunities for patenting things that are drug and device combos, because you're enrolling people with a device and you're treating them with a drug. So I think there are a lot of cool things you could do there. So it opens up a lot of new possibilities.
And I do agree with you that in Alzheimer we see this coming. It probably obviously opens up the question of benefit risk to treat patients that you don't know yet if they have a disease. Any quick thoughts on that?
Yeah, I think that's why I do think that the incentives are aligned here, because if you do your algorithm correctly, you're going to calibrate it so that you're getting people with a certain risk that's well above the cost-benefit ratio. Yeah. And you're going to see if you were right by looking at your control group. And so, everyone is incentivized to produce good algorithms that identify people who do end up developing the disease at the rates that you want them to. Not that you want them to develop, but that they are going to develop it.
Thank you, Ziad. This takes us one step further through the life cycle of R&D. Kate, we just spoke about, clinical symptoms and when to treat whom. Finding the right patients as inclusion-exclusion criteria for clinical trials. Where does AI help?
Yeah, I think I agree with the point of I would love a future where we get to prevention and we can actually, offset and prevent a disease from ever occurring. I think we're not there yet. And so if we think about like the oncology space, which is a space that Tempus works a lot in, and how those trials are run, even as we get better at identifying potential patients through diagnostics for trials, to really hone in on the right patient, right time, it creates a new problem of how do you find those patients. If you're going after a very niche patient, whether that's a genomic mutation or a gene signature, suddenly getting to those patients becomes very difficult. And so that's a place where I think AI, we heard a little bit about it this morning from some of the other panels, that we can start to use large data sets, EMR data, other types of real world data to identify those patients, especially when it's multimodal and it includes clinical data, genomic data, imaging data. We can really triangulate and find those patients and then hopefully pull them into the trials. We can talk later about some other problems that present themselves.
Yes, indeed.
But that's one. And I will just mention another really interesting application of AI is when you have a potential patient selection marker or a biomarker that you want to look at. We've also seen that we can actually create surrogates using AI for some of the more complicated biomarkers. We can reverse engineer and ask the question, instead of this really complicated gene signature, what other more common features could we utilize to be able to find those patients? And that's actually becoming quite a practical way to find patients. I think earlier today somebody was mentioning that even though NGS is no longer N, it's just GS, it's still incredibly hard to actually get all patients sequenced. So if we know that there's a surrogate marker for a mutation and we can find those patients, that's a practical application that's using AI.
So as sometimes signals, in particular in complex diseases, or signals of efficacy are not easy to find, can you just elaborate how is real-world evidence used in order to assess which patient subtype may respond? How does that work?
Yeah, I think this is another really fascinating area as we have more and more real-world data, and as that data is getting larger, we're able to actually sometimes completely model trials in silico. So to take a trial design and then go into a real-world data set and ask the question, if you ran this trial in these patients, what would the Kaplan-Meier curves look like? And we can do that repeatedly while modifying some of the inclusion-exclusion criteria that we just talked about to see if we can actually change the outcome of the trial in silico. And I think that's a particular application that can hopefully help us reduce a lot of the failure that we see in these studies today.
I'll stay on the protocol piece, and I'll then come to you, Ben, after that. So when you look at trial designs, how many head-to-head comparisons do we need that have the same comparator when certain, innovation products are getting tested? How can data and AI help to build shorter and maybe more ethical trials so that we don't need to do the control arms again and again and again? What's your perspective on that?
Yeah, I think there's a whole lot we could talk about here for a very long time. I think this is a place where actually the regulatory agencies could make a big difference as well, right? Because what you're speaking to or alluding to is that, right now we do a very, very linear model to clinical trials. I'll stick with oncology. I think it's the same in Alzheimer's and other disease areas, meaning that if you have a new drug and you want to show that it's efficacious, you have to run against a standard of care. And in some cases, the standard of care is changing so fast, which is a good thing. We're having a lot of great, amazing new drugs enter the marketplace. But oftentimes it's changing so fast that trials become outdated as soon as they've started. And so this other potential model that we could think about is really kind of Bayesian models, but where you're using real-world control arms that multiple companies could leverage, rather than each company running one by one their own study with their own control arm. Can we think about ways to start to leverage a single control arm that the regulatory agencies would accept, that every company could run against and share that data?
And I agree with you, and in my experience, the choice of control has a big impact on recruitment numbers as well, where in some countries it may be the thing to do as control, in others they're saying, that's no more standard of care for me. And so that's when you really struggle in your overall recruitment. Now, we spoke a lot about science decision-making, clinical decision-making, but let's take a step back and look at the technological opportunities, tier one, Ben. What opportunities do you define as tier one, and how can we get more efficient in everything around clinical trials?
Yeah, one of the biggest opportunities we think where the AI models are actually really good today is actually making a lot of the mundane tasks of what clinical stage drug development organizations have to do a lot more efficient. So think about things like biostats, medical writing, protocol development, some of these kind of things that take a lot of human hours and time. And then, moving beyond that over time, right, where then it's about decision support. So how do we basically give the best human decision makers in the company the right data to make the right decisions? And you think about the knowledge work and the bulk of what, that 70% of the cost is. It really is around the nuts and bolts of drug development and clinical trials. So we think the tier 2 applications are how do you make then better decisions? And throughout this process, you can begin to train kind of some of the large language models, and this is how kind of these LLMs are actually trained, is you ask like the best human decision makers to make explicit what's implicit. So why did you pick that endpoint? Why did you design the study that way? And that kind of chain of thought with the reasoning models, that's how you get to tier 3, where at some point the AI systems are making the decisions. And we kind of think that it's clear that the progression of how AI is going to make drug development a lot more efficient is first allowing one person to do 10x more work, right? Medical writing, that's probably intuitive, we kind of get that. Then it's around capturing why we made decisions as an organization. And at some point, then you're training this AI R&D scientist who will probably be able to synthesize a lot more information a lot better than any kind of single human can do it. And as a company, we kind of bet our kind of roadmap based off of this. Kind of our perspective is, drug discovery is only getting more and more efficient. There's a technology revolution creating more candidate drugs. No one truly knows what's going to work until you run a clinical trial. Per earlier sessions, that's where 70% of the costs are. So arguably, if you want one competitive advantage as a pharma company, it's the ability to run trials cheaper and faster. So as a company, we acquire and license clinical stage assets. We use this AI-enabled drug development engine to run the trials, and we look to sell after value inflection in the short term, back to pharma, one day kind of go end to end. But I think crucially, one of the things that we have the advantage around is change management, such a challenging dynamic here. And maybe just one example. So we recently publicly launched a tool. We have a big partnership with OpenAI, and Sanofi, where we built a patient recruitment tool called Muse. And traditionally, if we were to kind of develop a recruitment strategy, we'd have a pretty big team of drug developer, patient kind of recruitment experts, research the patient indication, segment the population, put together all the IRB-approved materials, the patient brochures, the ads in different languages, and make sure it's IRB reviewed. So you have this kind of compliance team take a look at it. It used to take our team months. And with the tool that we released, it's now minutes. That's pretty difficult for a lot of organizations to adopt, because if you're like a bigger company, it means that you need to essentially reorg to fully benefit from it. Because it's not a tier 1 tool, which allows one person to do 10x more work. It actually fundamentally changes the role and the jobs. And maybe just to call out, it can feel very existential for a lot of folks, in the companies. That's really where, OpenAI calls this level 5 AI systems, where AI systems become the org versus just the agentic kind of, it can do one thing kind of better. And we think that's going to be one of the hardest challenges, but also the greatest opportunities, where some of these systems are beginning to do the work of orgs. And you can apply Muse to putting together a TPP or a drug development program, right? So researching, understanding where the white space is. These AI systems are getting really good at doing that research and then putting together the right strategy.
So as you moved us into adoption, I have 2 questions. The first one is, so if the tier 1, the simple solution, the one person can do the job of 10, if that's more linear and easy, do you believe that drug development organizations all maximize these more simple, linear efficiency tools? And if not, why not?
I think that's probably where we're going to see the greatest adoption.
Yeah.
And usually associated with like point solutions, because in order to kind of capitalize on tier 2 and tier 3s, you do have to make a few kind of investments. One is around having just something that's not really sexy, but like a single data layer to capture all the different, multimodal data sets in your organization. Infrastructure can capture why people made decisions, so that kind of annotation. And then you're actually training and building your own models, and you're building also your own application layers on top of it. I think pharma as an industry is good at like buying a vendor. This is less about buying a vendor. It's about making an investment in inextricably linking how you operate, and how you operate, aka the application layer tools you're building, is actually training your AI model. So just to give an example, we have a medical writing tool. We pushed it in Word because meet the users where they are. As we make edits in Word, that's actually training our medical writing LLM, right? And that requires a bit of, the things are a little bit more challenging to adopt. So I think level 1's going to be, or tier 1's going to be easy.
Easier. Mm-hmm.
Tier 2, tier 3 is going to be a little bit harder. And tier 3 is the hardest because what you're asking an internal team member to do is train something that is going to take away their jobs. And so I think the friction for a lot of companies are like, and maybe just to, kind of, this is kind of an archetype that we think will play out is like, you have this new tool. The tool has the potential to drastically make your internal processes a lot more efficient. The end user is now required to assess that tool and see whether it helps to make their processes a lot more efficient. But if it's successful, they know like a 90% layoff is coming. And we think that's going to be a common kind of case that's going to make that tier 3 not only hard because of the org changes, but because of the human kind of elements that we're like very sympathetic for. As a company, we don't have that challenge because it's like we're just hiring and building the org with these tools in mind. We don't have to worry about maybe the 100,000 people who are hardworking, mission-driven. You want to find a role for them. I think that's going to be the existential challenge.
Thank you, Ben. And it touches what Martin mentioned. There is no digital adoption without an adjustment of analog workflows. Ziad, back to the clinical example that you brought in Alzheimer. Several diseases are ill-defined when it comes to treatment responses, in particular where we are earlier in the innovation space. And others take longer to show certain outcomes, such as overall survival, in particular in cancer patients that are less aggressive. So surrogate endpoints, what is your take on those and how can AI support?
Yeah, very positive on those things. And I just want to tease out, you mentioned so many important things, but when we think about the drug development landscape for cancer, we are very long on late stage cancer and very short on solutions for earlier and earlier stage cancer. Why is that? Because the trials are much harder to run. They take longer because you have to wait for cancer to develop. It's much more rare, so you've got huge populations. Yeah. So this is why the whole system is torqued towards this late stage treatment. And that's produced a lot of great stuff, and I don't want to diminish that. But we've also got a bunch of other white space in the earlier parts of the continuum that are currently not tractable because they're too risky and they're too expensive. So anything that gets that cost-benefit calculus to change is hugely valuable, and I think surrogate outcomes is one of them. So here's a very concrete example. In part because as a researcher, I find it really incredibly annoying to get data to do my research. I co-founded a company called Dandelion Health, which aggregates data from big non-academic medical centers, including imaging and waveform, ECGs, like everything, and makes that available for people who want to build AI products. So one of the things that we did was we got all of the electrocardiograms that were run on these huge populations of patients, and we ran an algorithm that converts your electrocardiogram into a well-calibrated estimate of your 10-year atherosclerotic cardiovascular disease risk. And so now you've got an electrocardiogram as an endpoint, which is pretty cool, because what we can do then is we can look at all the people who just started their GLP drugs, and we can see when they get an ECG, what happens to their risk. And we see in a year and a half, that risk dramatically dropping in parallel to what we found in clinical trials that took a lot longer than a year and a half. And so I think that's just one example of how this is going to completely change the calculus for doing this early stage preventive work. And it would be great if the FDA started approving these things. I think that's actually, as Ben and I were talking about last night, potentially on the table with the new FDA coming soon. But even if the FDA does not take these things seriously, those kinds of surrogates are really useful for internal decision making about which compounds get moved forward and which don't. You can do a lot of real world evidence generation just from looking at treatment initiation of similar drugs or things like that. So I think that this has the potential to really transform not just the length and the size of clinical trials, but the kinds of drugs that we can test in a risk neutral way.
Thank you so much.
Yeah.
I will touch, sorry, please.
I was just going to hop on that bandwagon for 1 second, because I do think that MRD in oncology is a great example of what you're talking about. Surrogate endpoint, moving disease earlier in the neoadjuvant and adjuvant space. And actually, it took almost a decade, but the FDA has now accepted it just recently, last week, as to be able to look at those patients and changes in this case, ctDNA, which is not AI yet. But we know that if we can use that and we can start to label in additional features, it will very quickly potentially be a next wave coming. So just wanted to add to that.
Thank you. We have not touched one topic yet, which is critical. And Jeff, I'll ask you to give us your first take on it. And please think of your questions, because we are getting towards the end. Clinical trial execution is maybe the thing that takes the longest and costs the most. Where can solutions help to find the patients and accelerate trials?
So for one, I think AI is going to be out throughout the entire clinical development process. I think it's been pointed out here, it's a heavy administrative process, both for the clinical sites and actually for the sponsors.
Yes.
We probably haven't made a clear enough distinction between what occurs at the site, the research site, versus what the sponsor activity is. But within the workflow of the sites, at least what we're beginning to see is almost AI is going to be used to identify the patients that meet the eligibility as just a standard part of how they operate. They can't hire enough people. They can't retain enough people. It actually tends to be a cost center, not a line that contributes positive margin. So its efficiency, its effectiveness is super important at that level, and there's no barrier to adoption. In fact, we're now finding whole networks actually going 100% AI in their workflows and planning on doing it as new trials and activities come into place. The ability to collect that data can also be fully automated. So we're already using large language models to pre-populate the electronic case report forms. There's no reason to take electronic available data, actually manually key it in while looking at a CRT where it's electronic, and then get it back into an electronic form just so it can be written to a backend EDC. That will actually start to disappear, particularly these models have the accuracy of the research. Now the research team still has to confirm that, actually review that, et cetera. And most of these things have human reconfirmation throughout, but you're taking things that took hours and getting it down to 15 minutes. And that's actually going to add a lot. When you start adding that all up and also adding that a sort of analytic ready data set can be available with no source data verification process being required, which is what's actually happening on the studies where we're deploying these technologies today, you can start seeing we're going to take out 30 to 40% of the actual cost and time of this whole consideration all the way through. If you go earlier studies, actually continuous analysis of phase 1 is possible. Back to this point where these are semi-Bayesian, they've got branching logic, and actually you can terminate earlier, you can advance to the next branch earlier, so the whole approach to early stage is going to start changing. That's already going into place as well.
Thank you. Kate, diversity in clinical trials, how can AI help in the execution?
Well, this is another really big topic. I will say, just adding to what was just being described, we can find patients, we know where they are, and it's interesting, I think we heard the theme this morning in several of the panels that last mile of actually deploying and implementing is a really big challenge. And I think this speaks to a problem that we have with, really getting diverse populations into our studies. So we know that it really comes down to, you can have the AI, you can find the patients, it's then getting those patients into the studies. And sometimes that takes boots on the ground to do kind of two big things. One is to really work with the physicians at those sites, and two is to really work with patients to get them interested in the study, to explain the study to them, and sometimes to solve really intractable problems that patients can be dealing with in particular in different socioeconomic areas, which is sometimes things as simple as transportation to the site. Or if the clinical trial is running at a site that's not near their home, how do they actually access the site? So I think AI is able to help us in ways, but then uncover new problems that we have to find solutions for.
I agree with you, and I've been involved in successful clinical trial accelerations where we actually found that the activity that was most impactful to accelerate active recruitment of sites was face-to-face visits, and with good, impactful field teams or scientists. So that's where the analog and the digital balance find each other again. So we have time for one or two questions. Clinical trial, there is someone with a question. Sorry, I don't see it well. It's such a big topic. There's another one.
Thank you. Well, my name is Parker. I got squeezed two in. I'm hearing that there's more and more concern about surrogate endpoints. I understand the enthusiasm for them, but there seems to be quite a lot of evidence that actually 5-year total overall survival is not tracking these surrogate endpoints, and big pharma are moving away from it. That really worries me, because that seems to be something that would drive up the cost. So I'd love to hear about that. And then if we have time, Kate, I loved your point about the platform trials and simulation, Bayesian adaptive trials. It seems the big challenge, there's a great one here in UCSF with iSpy, but you need to have a kind of organization, normally a not-for-profit, to intermediate that. And so there seems to be like a governance barrier to that happening. Any thoughts on how that might happen?
I can start on the first one. And I think the current way that we develop these surrogate outcomes is a very, I'd say, human process, where we have in our heads a causal model of like, here's the chain of events that leads to the ultimate outcome. And I'm just going to move up the chain a little bit. And I think that's, as often as not, illustrated how little we understand about the disease that we're studying. For example, many drugs reduce blood sugar, but increase diabetic complications and mortality. So I think there's a lot of problems with our current way of doing surrogate outcomes. But notice that that's all a statement about the human process of developing these outcomes. And I think when we have algorithmic approaches to developing those outcomes, of course there are different sets of problems that can come up, but I think that that's generally a much more rigorous strategy of finding these correlations in the data that will track and will respond to the treatment just as well.
Thank you. Kate?
Yeah, and maybe to speak to the second question that you had, I think, I agree with you. In the end, I think in order to really be able to implement this kind of Bayesian, shared control arm, shared real-world data strategy, which would speed things up and benefit patients and create more innovation, I think it will come down to consortiums and large groups coming together and being willing to share data and work together. But we have a lot of precedents for that, right? We talked this morning about the Human Cell Atlas is a good example. I know when I was in the pharma world, we just spoke about MRD. That was actually an example where in heme-onc, numerous pharmaceutical companies, academic institutions, and large consortiums came together to generate the data, share data, to be able to ultimately convince the FDA that was a legitimate surrogate endpoint, to come together around methodology and standardization of how to collect the data in trials. So I think we're going to see, or need to see, similar approaches now with utilizing AI in clinical trials.
Thank you. JC, you have a question.
We have been using, with mixed results, synthetic arms, mainly because probably humans were really generating them. What do you think that is the potential of synthetic arms going forward? And how do you ground truth them as you go forward?
Well, yeah, either way. So there's synthetic, and then there's what's also called external control arms. External control arms would be derived from real-world data, roughly contemporaneously collected, and particularly in diseases like cancer, where if the current standard of care is completely inadequate, it's a very rare population. And if you've got a therapeutic entity that has promise, you want to put that person on the active kind of area while you're kind of creating an external control arm. Those have been accepted. They're actually accepted both by European authorities, US authorities, and we've been part of numerous drug approvals where those have gone all the way through to approval. And so I think those will continue to have high viability. You want to make sure, though, they are truly as representative as the actual population that's actually being brought into the trial. But these are small numbers. You're talking about a trial where there's 250 to 500 subjects at the end of the day, even for a kind of a mid-stage trial. But that's a very viable area.
I'd say there's a ton of, just in the past 3 to 5 years, really interesting new methods for creating different kinds of synthetic control arms. And I think one of the things that's been missing is the data in which to test these kinds of things. And so I think as more and more high quality electronic health record data from health systems comes online, and we get richer and richer ways to create those synthetic cohorts via imaging and years of longitudinal multimodal data, I actually think that those things are very promising. We'll see how they work ultimately, but
thank you so much. Thank you to the audience as well. This was a fantastic panel. I learned a lot. Thank you so much.